What makes you the unique person you are? Maybe it's your smile, maybe it's your kindness. From a biological perspective, there's a pretty simple answer to this question of what makes you the person that you are. It's DNA. There's a longer version of this answer too. That is deoxyribonucleic acid, the same as DNA. DNA is a critical molecule that makes up exactly who we uniquely are. DNA has a unique structure of a double helix. A helix is this gray piece here, and it has two helixes or helices, so we call it a double helix. On this double helix are combinations of four nucleobases. We have thymine in red, cytosine in blue, adenine in yellow, and guanine in green. The order of these bases code for particular amino acids, which make up proteins which do things in our cells. Not all of the bases can code to one another, can pair with one another. So thymine and adenine always pair together, and cytosine and guanine always pair together. They make a triple bond. Adenine and thymine make a double bond. And so if you have one strand of DNA, then you can match it and understand what the other strand would look like, because there's only one option of what it could look like. As I said, these base pairs then code for amino acids, which is part of the central dogma, which we'll talk about in a little bit. And these proteins then make up every single thing that a cell does. So this week, as you might have guessed, we're talking about heredity and inheritance. There are similar words for the same phenomenon. It's how does genetic material transfer between generations? So what do you get genetically from your parents? What did they get from their parents? How are they unique? How do they uniquely combine in every person to create a unique individual and a unique set of circumstances? So what does DNA actually do? I've gotten at this a little bit. So I think most people probably know that DNA is a genetic molecule. It carries our genetic information between generations. However, in order to do this, it converts into RNA in a process called transcription. And this works because, as we said, there are two strands of DNA. It's a double helix. And since each of the bases can only bind to one other base, then in transcription, the DNA is unzipped. So you're only working with one strand of DNA, and RNA then is coded onto the strand because it can match the existing strand into a new molecule called RNA, which is only one strand, but matches DNA. So it reads from the DNA and creates this new molecule, which then, in the process of translation, can become a protein. So reading 3 nucleotide bases at a time, then the RNA can convert to amino acids, which are the building blocks of protein. And these building blocks of protein then create full proteins, and proteins are the doers in the body. They're the molecule that creates pumps that go into cells and creates proteins, creates chains of carbohydrates, all kinds of the cellular mechanisms in the body are actually proteins. And so when you get proteins, then the body can code for exactly what it needs to do, all the things that it needs to do. And so in this way, DNA that is passed from your parents becomes protein that does all of the cellular mechanisms of the body. So this is the central dogma, that DNA, through the process of transcription, becomes RNA. RNA acts as a template in order to code the amino acids of protein in the process of translation, and proteins then make up all of the important things that do anything, all of the important doers in the body. So central dogma, DNA into RNA into protein. This is really critical because this is how genetic information makes us who we are and do what we need to do. So how does DNA pass between generations of cells? So if you're talking within the same person, so say you took a cell from a muscle in your leg, so just one single cell, that cell has all of the DNA in your body. Every single cell has all of the DNA in your body. And so, how does that pass a DNA to a new muscle cell that's being made? So say you're working out, your muscle cell is getting older, it's getting tired, and needs to make a new muscle cell. And how's the DNA going to get into that new muscle cell? So this is through the process of mitosis, which you might recognize. It happens in the larger context of the cell cycle, this creation of new cells. The point of mitosis is that there's very little variation between parent and daughter cells, because as long as it's working correctly, the muscle cell in your leg just needs to make another version of itself in order to continue having this strong muscle in your leg. And so this only works for somatic cells, which is cells that are not sex cells. So we're going to talk a little bit about sex cells, which are also called gametes. So our ova in females and sperm in males. And so this is how every single cell in your body that's not one of those two replicates. So you start and end with 2N or diploid cells. So what we mean by diploid is that there are two sets of chromosomes. and you end with 1, 2 sets of chromosomes. These are the same. This is a little confusing, but before prophase happens, then this envelope, the nuclear envelope, dissolves, and the chromosomes condense from 1 into 2 segments like this. So these actually are exactly the same. They just don't look like it here. So in the step of prophase, as we said, the nuclear envelope dissolves, and you end up with 2 arms in chromosomes like this. Chromosomes often form an X shape. And this is because at the step of metaphase, the chromosomes, all the chromosomes in your body, in your cell, will line up at the center of the cell, just like this. Then these fibers in anaphase will attach and begin to pull them apart into the 2 different halves of the cell. So then in telophase, you have the reformation of this nuclear envelope and bringing these cells into 2 individual halves. And then the cytoplasm, this is all of the stuff floating outside the nucleus, splits, and you have 2 identical cells. So it's critical that there's little variation between these cells and that you start and end with diploid cells. You start with the same number of chromosomes as you end with. However, DNA can't pass in between generations of people in the same way. Otherwise, we'd all be running around like clones. Everyone would look exactly the same and have the exact same DNA makeup. And of course, we know that's not the case, that people have moms and dads that contribute half and half to their DNA. And so the way that this works is you start with exactly the same. You start with a diploid cell, like here, and you end up with haploid cells. So we're going to walk through this again. This half, so meiosis 1, looks a lot like mitosis, what we just ran through. And so just for the sake of illustration, we're starting off with 2 sets of chromosomes. So you have the blue set and the pink set, and those are then going to cross in our sex cell. So either in our ovum or in our sperm. This crossing is why people are not exactly 1/2 replications of their parents. There's some intermingling of those traits in this step called crossing over, which leads to genetic diversity. So then in anaphase, the spindle fibers then pull apart the chromosomes, and you end up with telophase looking a lot like our first version of telophase in mitosis, and split up into these 2 cells now in cytokinesis. You split the cytoplasm. And then from here, we have these cells that have already crossed over, And so they look a little bit different than the ones that we started with. And there's also only 2 chromosomes here in this example. So then basically the same thing happens where you start with prophase, dissolve the nuclear envelope, attach the spindle fibers to the center of the chromosomes. They pull into sister chromatids. These then reform the nuclear envelope, formed into each having haploid cells that are divided in cytokinesis. So if you can see, these cells each have half of the chromosomes that we started with. They also are a mixture of the cells, the chromosomes that we started with, because of this crossing over step in metaphase 1. So we have half of the number of chromosomes, because otherwise, if we were to do mitosis and you would try to bring 2 diploid cells together, then they would double in chromosome number, right? So you would end up with 4 sets of chromosomes and then 8 sets of chromosomes, and so on and so on. And so this is how, as humans and other types of animals, we end up with the same number of chromosomes between generations. Not all organisms have 2 sets of chromosomes. So bananas, for instance, have 3 sets of chromosomes. Genetically modified corn might have up to like 50 sets of chromosomes. So having 2 sets of chromosomes is a particularity to being human and a lot of other types of mammals.